Welcome to Neomatician webinar series, where academia and industry experts converge to exchange ideas, engage in thought-provoking discussions, and seize networking opportunities. Neomatician offer a diverse range of courses, organize captivating webinars, foster collaboration, and promote continuous learning and knowledge sharing. Our aim is to cultivate the next generation of technologists and innovators. Lead by our host Dr. Preeti Prasad to ensure that we make the most of our time together. Let's welcome our guest speaker. Dr. Hitesh Verma is a seasoned peptide expert in pharmaceutical industry. He brings a wealth of experience to our virtual stage as the Senior Project Manager at Boehringer Ingelheim in Germany. With an impressive career across various esteemed roles such as Deputy Manager at Alembic Pharmaceuticals in India and Assistant Manager at Aurobindo Pharma, Dr. Verma has a deep understanding of the pharmaceutical industry, notably Dr. Hitesh Verma holds degrees from prestigious institutions including École Polytechnique Fédérale de Lausanne in Switzerland and a PhD from Indian Institute of Science. He will share with us his expertise and insights in peptides to pharmaceuticals, the cross-continental journey from laboratory to leadership. Please join me in extending a warm welcome to Dr. Hitesh Verma.
So then I joined some industries. Initially, I joined in Alembic Pharmaceutical, and later on I moved to Aurobindo Pharmaceutical. Now I'm working in Boehringer Ingelheim as a senior project manager. And like all my expertise is in peptide, like my PhD, my postdoc, my industry experience, everything is peptide. So I'll explain you what I have learned so far, and that knowledge I'll share with you. And if in case you have questions, I'll answer at the end. Okay. So let's begin. So like peptide is a very you can say a novel modality that is being now very popular. It's been few years now that people are talking a lot about peptide. Companies are investing huge amount of their budget towards peptide development, and it's not just peptide. It's like nucleotides as well. So commonly we call it tides because we can refer to both of them, peptides and nucleotides. So here is a like huge investment coming in the field of peptides and nucleotides. And why is it so? Why it is getting interested, and why everyone, like each and every company or academicians also, are focusing on peptides? So that we'll discuss today. So starting with certain things, like I want to show you like there are different chemical modalities which are available. Okay, people actually work on small molecule, which was a forte of research for a very long time. People used to study small molecule for drug development, for drug discovery, for developing an inhibitor or a blocker or something like that. And generally, small molecules are in the range of 500 to maximum of 1,000 Daltons. But then like after that, a huge gap came. Like people started working on biologics and enzymes. Okay, and then generally biologics are like big. You can say monoclonal antibodies or large protein molecule, which are very high in size, like more than 150 kDa or something like that. And there was a gap. Like very few people were working on a gap where we have molecules which are in the range of 1,000 to 10,000 Daltons. So why did people actually, focusing on small molecule and neglecting a gap and then working on biologics? So it's a huge difference. So small molecules are very good inhibitors. You can say if you have any enzyme, these small molecules are very good in going to pocket and binding there and blocking the activity. Like and if we talk about biologics, so there are many antibodies which are available, which are being used as a therapeutic moiety. But then came a need of these molecules which are in between, like ranging from 1,000 Dalton to 10,000 Dalton. So reason for this was that as the uh, field of biotechnology evolved, people started working on more of different pathways, different proteins, different mechanisms, And they came across a plethora of proteomic proteins, or you can say transcriptomics, which was available. But unfortunately, all proteins or all, what do you say, these receptors were not having pockets which were similar to those where small molecule can bind. So then this term came in like initial of 2000, that there are proteins which are undruggable. Like we cannot target them with a drug. The reason being that these protein has larger surface area, which cannot be blocked just by small molecule. And also these proteins have certain sites that can be recognized by antibodies, but those sites are similar to what we have in natural proteins as well. So these proteins then became undruggable in the sense because they have a larger surface area that needs to be blocked, and antibodies cannot be an alternative for that. So then that people started coming with these modalities, which are peptides and nucleotides. So like on slide, I'm showing you now how the peptide drugs evolved. Like in the next slide, also I'll show you how like peptides evolved in 1920s and all. But I'm specifically showing you recent years, like 2016 to 2018, and data from 2016 to 2023. So on this slide, you will see how many drugs, total novel drugs got approved in 2016 to 2018. So actually this era, the 2017 was the golden era, which FDA also calls a golden era of drug discovery, because many drugs got approved. I don't remember the exact number, but it was in 56 or 54 number of different novel drugs got approved. And out of these, whatever number of drugs, a considerable amount of drugs were falling in the category of peptides. And eventually, when the research went more in these area, we observed that there was number increase in the number of the peptide drugs getting approved. So earlier, people were reluctant towards studying peptides, and there are reasons why people were not studying peptides or nucleotides, because they get degraded easily. You cannot have a oral peptide or a oral nucleotide, because again the degradation is my main challenge. The permeability of these category of drugs is not that great as we talk about small molecule, but they have their own advantages. As I mentioned, the targeting of undruggable protein is one of the major advantage that we have. So yeah, as I was talking about the evolution of these drugs, you can observe that first the peptide drug which we call was the insulin. Many peptide chemists, they differ with this point, because many people argue that insulin is a mini protein and not a peptide. But this is a topic of debate. But peptide chemists love to call it that insulin is one of the peptide drugs which got approved in early 1920s or so. And during those course of time, the peptide field was actually evolving. You can see, like in 1954, there was first time oxytocin, which was a small peptide, which was being synthesized chemically in the lab. And it is not easy to synthesize peptide. Those who understand peptide, they know that peptide synthesis is a lengthy process. It involves multiple steps of synthesis. So we thank a lot to Nobel laureate Merrifield, who actually discovered SPPS, solid phase peptide synthesis, and that made the life of peptide synthesis or the discovery of peptides very easy. So solid phase peptide synthesis, the discovery of this particular process was very important. It was a milestone in the field of peptide discovery, because it gave a path that how peptides can be synthesized sequentially, and how a chemist can do various modification and thereby can make it to be a drug molecule or something like that. Then after that, many peptides came into the market. You can see a list of peptides which I have shown here, but there are many peptide drugs which got approved. Very recently, you can see over here, I'll convert it into a pointer, I would say. Yeah, so you can see over here recently, liraglutide, linaclotide, dulaglutide, semaglutide. These are the drugs which are covering most of the peptide drug market. And these are the recent, you can say, milestones which are being achieved by various companies in getting approval. And I'll talk about these drugs more in the next slides. So next, I would like to tell you about what are the therapeutic areas where peptide drugs are being, used nowadays. So it is not limited to certain condition nowadays. Although there are like mostly peptide drugs are available in market which are against oncology or metabolic integration indications and endocrinology, because like we know many hormones which are there in our body are actually peptide hormone. So it is easy to utilize various hormones and use them as a drug. Metabolic integration, because as I mentioned, insulin was one of the peptide which is used as a drug. So there are various other metabolic condition that can be controlled by just the modulation of insulin also in the body. And there are other indication as well which are now getting picked up and now getting peptide drugs are getting approved in these conditions. So as you can see, most of the peptides are agonist. So you know the difference between agonist, antagonist, and other categories of drugs which are available in the market. So this is how like the current you can say distribution of peptide drugs is there in the market. But eventually, I'm sure in next few years we'll see many more drugs covering many aspects of therapeutic indication in the market. So to explain my point that like how peptides are being moved from laboratory to pharmaceutical, what we have decided to be as a topic of our discussion today. So for that purpose, I have made a case study. So I'll be talking about one of the anti-obesity drug discovery, like how this peptide got discovered for a particular condition. And I have chosen obesity specifically because obesity is an indication which has picked up recently. Like it is being a point of focus for many industry, many companies, because obesity is involved in various other condition as well. Like many people who are obese have metabolic problems like diabetes, fatty liver diseases. So many other you can say therapeutic condition or indications are associated with obesity. So the obesity becomes a very you can say a soft target, or you can say a very high demanding target right now. And peptides are doing really good in the field of obesity. So it's not like there are no drugs available for obesity so far. There are many drugs which are being in market and which are being used to treat obesity. And they are the various they are against various targets. Some are mitochondrial uncoupler, some are they block certain receptors, there are inhibitors, there are agonists. So various targets are there. These are small molecules and many of them are still under use, like you can see this one. So many of these drugs are still in the use in various markets. But I want to take your focus on this particular thing, GLP-1 receptor agonist. So liraglutide and semaglutide, these are the recent peptides which got approved, one in 2014 and one in 2021. They were specifically approved initially for type 2 diabetes, but then people saw their uh, activity against obesity as well in the process of weight reduction, and now they are approved as a marketed drug for against obesity as well. So I'm not going to talk about these two drugs, but there is one drug which is specifically taking focus of various companies. So that I'll talk about. Now, before that, I want to tell you like what I'll be talking mostly in my slide. So it will be the life cycle of these drugs, how the drug discovery happens. So initially, the target identification, how a particular target is getting identified, whether it is a druggable target or a non-druggable target. So that identification, generally mostly these biotechnologists or various biochemists, they perform research in identifying a protein or a particular biologic which is involved in a pathway. And when it gets blocked, the condition or the disease can get cured. So this is the thought process that goes under target identification. Then once the target is identified, we use various approaches to identify a hit. We use various approaches to design an agonist or an antagonist or any molecule that binds to that particular protein or whatever, and cure the disease. And then when that hit is identified, that hit is optimized to get a better molecule. Like many a times, the one which is the better binder is not stable enough. The one which is a better binder might not be, can have immunogenic effect or mutagenic effect. So that optimization is necessary. So lead optimization happens, and once the lead is optimized, people perform preclinical trials, clinical trials, and ultimately they go for submission, and finally the drug gets approved. So over here, I'll be talking about a drug which is tirzepatide, and tirzepatide is a drug which is being approved by like Eli Lilly, a very well-known company in the field of drug discovery, and it is a dual what do you call agonist. So it works in by targeting two different moieties. So I'll just explain you a bit about what are incretins and this tirzepatide, how it is derived from incretin. So there are two hormones which are present in our body. One is GIP, another one is GLP-1. Okay, and these two hormones are very important hormones which help in maintaining the homeostasis in the body. A GIP hormone is a 42 amino acid long peptide, and it is synthesized as a 153 amino acid peptide, gets cleaved, and finally we have a 42 amino acid long peptide, which is the active peptide. Also, GLP-1, glucagon, glucagon-like peptide. Initially, it is synthesized as a proglucagon, 160 amino acid, and once getting cleaved, it has a 31 amino acid long sequence, which is the actual hormone which do the activity of GLP-1. Now, this GIP and GLP-1 are mostly involved in increasing the insulin level. In fact, GIP is also involved in increasing the glucagon level, while GLP-1 is involved in the reducing of glucagon level. And they perform various other activities as well. So they have a very important role, you can say, in maintaining the homeostasis or the physiological condition of the body. Now, once when we take the nutrient in, once the food gets in, the K cell and the L cell, which are responsible for the secretion of GIP and GLP respectively, they synthesize these peptides, and these peptides do the positive incretin effect. Now, incretin effect is nothing but the effect related to the insulin secretion and the performance of the insulins. So these hormones are responsible for the secretion of insulin, and later on, whatever insulin does, like assimilation of the food, glucose, and all other activity. But unfortunately, these hormones are not highly stable. Like you can see, the half-life of GIP is 4 to 5 minutes, and half-life of GLP-1 is just 2 to 3 minutes. And there is a protease, DPP4 protease, which actually cleave these two peptides and make them inactive. And these are the degraded product, which are not active. And finally, they get cleared from the renal secretion. So though our body produces these peptides, but they are producers just close to the site of the action. They perform the action and gets cleared off. So their physiological levels are very low. They are not produced in very high quantity because their activity is just to the localized tissue only. They travel to that tissue, perform their activity, and gets cleared off. But these two are very important hormones, and they can be utilized in maintaining the blood sugar level and ultimately the obesity and other thing. So these two hormones are individually being used. The drug which I was telling you earlier, liraglutide and semaglutide, they are actually a derivative of GLP-1 hormone. So those are nothing but GLP-1 hormone modified in a way that they are more stable, more active, and thereby they have similar activity what GLP-1 causes. So those were the already discovered drugs that are there in the market. And like liraglutide is one which is given frequently, but semaglutide is one which is administered once in a weekly dose. Okay, so you don't have to take the injection or the hormone every day. It's like once you take, you're good to go for a week. So the stability you can see instead of 2 to 3 minutes, the stability has been increased to multiple days. Okay. Now, why did we needed, like if we already had semaglutide, why the market needed tirzepatide, or why there is a need of another drug? Now, as I mentioned, that there are two hormones, GIP and GLP-1. If both of them can perform the activity, so obviously we will have a better acting molecule from what we had earlier. This was the thought process. So people started studying these peptides or making combination of these peptides and come up with a single peptide who can perform both the activities. So this was the first you can say study which was published in Science Translational Medicine in 2013, where they made an attempt to design a unimolecular dual incretin which can perform these metabolic benefits in rodents, monkeys, and humans. So again, unimolecular, just a single molecule that can perform both the activities of GIP and GLP-1. Okay, so they came up with a list of peptide. You can see there is a glucagon, GIP, GLP-1. These are the sequences. And there was one, this exenatide peptide, which was again a approved drug, liraglutide approved drug. And they came up with a list of peptides that let's make all these peptides and see which of these peptides act better against different receptors, GLP-1 receptor, GIPR receptor, and glucagon receptor. Our purpose is that we should not induce glucagon much, but we should have a better acting molecule, which is GLP-1 receptor binder and a GIP receptor binder. So they got this 20 number peptide, which was a very good binder for GIP and GLP-1 receptor, but not that good binder for glucagon, which is a very good molecule for us. And they published this paper saying that peptide 20 is a very, very good peptide and can be utilized for therapeutic purposes. They did some experiment on rodents as well and proved their point. Now, from there, Lilly actually took this challenge, and obviously they would have performed various studies, so they could publish in 2018. So they came up with a molecule which was very similar to peptide 20, and they named this molecule LY3298176. So this is the drug which is actually the tirzepatide. So earlier was a hit identification, and now Lilly actually did the lead optimization, you can say, and they came up with a very good acting or a very better molecule, which was LY3298176, which is a novel dual GIP and GLP-1 receptor agonist, which can act against type 2 diabetes. This was their first claim, and it was published in 2018, and simultaneously the clinical trials also started later on. So you can see over here, this was the peptide 20, and this was the optimized tirzepatide peptide. I have shown a sequence similarity so you can understand how different this peptide is from the actual hormone and how different this peptide is from the peptide which was discovered by the academic group. Indiana University and German group, they were the academic group. They published this paper, peptide 20. And the tirzepatide has 43% of GIP content, 40% around of GLP-1 content, and there are some common amino acids which are present. So this is a very interesting peptide, which they named finally as tirzepatide, and I'll tell you what this peptide can do and why it is very, very interesting nowadays. So Lilly started with clinical trials. So they started with two different clinical trials. One was the Surpass, another one was Surmount. So Surpass was the clinical trial to prove the efficacy of this tirzepatide against type 2 diabetes. So the objective, obviously in type 2 diabetes, the objective is just to lower the blood glucose level and maintain the homeostasis. Okay, and there are many trials which were being designed to cater different population, like those people who are taking metformin, those people who have metformin and have this SGLT2 inhibitor and all. And this is a list of like drug trials which generally every company go through when they will come up with the anti sorry diabetic drug. Okay, so some of the results have been published. You can go through those results. I'll discuss some of the result over here. But if you want to read more, you can just go through and search for these clinical trials, and you can have the data. So here I'm just mentioning about the brief about the surpass result. So over here you can see that initially they went ahead with different dosages of the tirzepatide and compared with the semaglutide. Semaglutide is the one drug which I was mentioning, which was approved in 2021 for type 2 diabetes. Okay, and they observed that there is indeed a change in the glucose level, percentage glucose level, and that was the effect that they observed, that like how the glycated hemoglobin HbA1c level that is being reduced and how it is being reduced at the in due course of time. So now I would like to make a point over here why it is important to perform these trials for so long, like for 40 weeks they had to perform the activity and understand, because many of these drugs when get administered in the body. So our receptors, okay, you can say, get used to it and then stop responding to these receptors or these ligands. Okay, so there occurs a stage when our body will not eventually respond to insulin or to these GLP-1 agonistin also. So there is a need that how long these drugs can be administered, and then later on will have a static effect. You can see there is a static effect, and then no further change can be observed also. So after that, then there is a need of change in the medication or the dosage or all those things that had to be decided by the physician. Okay, so yeah, that was the reason why people do this trial for that long. Again, the people who are responding to this drug, like those people who are having less than 7 of the HbA1c count or less than 6.5 or 5.7. So huge population was responding to this particular drug. And no doubt it was a very effective drug. The glucose level were reducing. It was around 100 for many people, and it was maintaining for a long period of time. Also, they observed change in the body weight. So compared to before drug and after 40 weeks, people observed there is a considerable amount of weight loss which was happening because of the drug, and thereby they decided that okay, we'll go for anti-obesity drug as well. So initially, the focus of this particular drug is just anti-diabetic, but once they observed these phenomenon or these results, they wanted to go ahead with the anti-obesity trial as well. You can see that cholesterol levels were also increasing. So overall, the activity of whatever GIP and GLP-1, because GIP is actually responsible for reducing the cholesterol level, maintaining the adipose cells tissue level, and all those things. So that effect was also observed. So then Lilly came up with the requirement of surmount clinical trial. Surmounts are those trials where mostly obesity was considered as condition instead of type 2 diabetes. So there are many trials, like one of them was just people who have BMI more than 27. They can participate in the activity. Other who has diabetes and also has BMI more than 27, and thereafter many different trials with different combination of drugs and different conditions like cardiovascular diseases are there. So what would be the response of the people and all those things, and also in different chronic kidney condition and all, because like all these conditions are actually, or diseases are correlated. Obesity, as I mentioned in my earlier slide as well, that obesity leads to cardiovascular disease, leads to kidney diseases, leads to various other metabolic conditions, NASH, MASH, all those conditions which are liver associated. So it becomes very important that these peptides or these drugs should act against those as well. And thereby, people like companies perform various trials which are against obesity, which are against cardiovascular diseases, which are against liver diseases. So this is the result of the anti-obesity clinical trial. You can see with placebo, there was not much of the reduction in the body weight. But when tirzepatide was injected, you can see a dose-dependent decrease in the level of the body weight. And Terzepatide was indeed able to reduce the weight to 20%, and 20% is huge. If you have a person with 100 kg body weight, he can attain the body weight of 80 kg in 40 weeks or so. So that's what this drug is capable of. So that's what they have performed these trials, finally got the approval. You can see the clinical trial data. The minimum they could reduce was around 15%, and mostly they could attain in 60 to 30 patient around the reduction of around 20% of body weight, which is a huge success for them. So I'll just tell you what was the timeline or what was the journey of this particular drug, because as we were discussing about like peptide, how it was being discovered and how it got approved finally. So in 2013, the Indiana University and German University people came up with the idea of unimolecular targeting, where they have the combination of GIP and GLP-1 agonist together. So that was like we can call it as a hit identification. Finally, Lilly published the paper where they came up with the lead optimization and LY32916. So that number, that drug, which was being later called as tirzepatide, uh, was as the lead molecule from Lilly. And then they started performing various trials from 2019 to like till date, the trials are ongoing, and they got the result in like timelines I have mentioned in 2020, Feb 21, Jan 22. So there are these, like I mentioned about these different clinical trials, which are there, Surpass and Surmount. And finally in 2022, 13 May 2022, FDA announced that tirzepatide, whose brand name is now Monjaro, is approved for the treatment of type 2 diabetes. And later on, after 1 year, FDA again announced that Zepbound, which is a formulation for tirzepatide, can be used against chronic weight management, which is like um, weight, which like people who has a body mass index of around more than 25 or more than 30, like that. And still many clinical trials are ongoing, where we eventually will get to know about what are the effect of this drug against cardiovascular diseases or chronic kidney diseases or MASH or other liver associated diseases. So like the on paper on slides, you observe that it is very easy, to have a peptide drug, to have a particular like hit identification, lead optimization, and then clinical trials and everything. But it is a lengthy process. You would have observed that 2013 was the first paper published, and now to 2022 or 23, or almost after 10 years, we got the approval of the drug. Okay, so FDA has a path or a process which every company or every drug has to follow, whether it's a small molecule, whether it's a peptide, or whatever it may be. And these are like specifically or strictly regulated guidelines which are there in which are available in the FDA website and other websites. So initially you have to develop a drug, you have to perform some animal test, and then you have to file a IND application. Okay, so IND is nothing but investigational new drug application, which every novel company or every drug discovery company has to file whenever they come up with a molecule, that we have a molecule and we want to go for human trials or clinical trials. And once FDA assesses your application, says that, yeah, okay, it is safe to be administered in human based on animal result, then various clinical trials like phase 1, phase 2, and phase 3 clinical trials are being performed. Phase 1 trials generally involve few people, healthy people, and we administer the drug in healthy people and see how this drug is being managed in healthy population. They are able to clear it or not. There are no side effects associated with these drug. And once the healthy people respond well to this drug, then comes the patients. Like many number of patients get recruited, varying in the age, gender, lifestyle, and then they are recruited for the phase 2 clinical trial. And once we get good results from phase 2 trial, then in phase 3 trial, we get larger number, larger variety of people, larger like people can have type 2 diabetes, people can have combination of diseases, and then how these people respond to these drugs. And eventually the drug will get approved or in the market. Okay. So like in approval process also, we need to prove what would be the drug labeling, and there is like we can go for 1 hour on talking about what drug labeling is and all those things. And once the drug is, you know, you can say that you have all the positive results, you apply for NDA application. So NDA is the actual application which is approved and which shows that your drug is safe for market. And once the NDA application is approved, people buy the drug in market, and then eventually phase 4 trial starts, like where you understand how the drug is responding in the overall population, and if something or some uh, side effect or any unfortunate incidents is happening. So those things are reviewed in phase 4 trials. Now, again, like peptide is being new in the field. So I hope I'm not crossing the time, Preeti.
No, Dr. Hitesh, please continue. It's looking very
Okay, okay,
interesting topic. Yeah, please continue.
okay. Yeah, because I'm just talking.
No, it's
like,
very
okay,
interesting,
okay.
and I have very new knowledge. I'm getting very new knowledge from you. It's a very interesting talk you have. Please continue.
Yeah. So yeah, uh, as we observed that there are many young kids from kids to nahi bolenge, हाँ, but young guys and girls are there in our audience. So I just wanted to show them the difference between drug substance and drug product, because many PhDs or many science students, they don't understand what is the difference between drug substance and drug product. So whenever you see a drug in the market, you just have to see what is the ingredient present in that drug. So you can see over here, many drugs actually have this particular thing, that what that particular bottle contains or what that tablet contains. So initially the first one, which is the, like in this condition, terzepatide, is our active pharmaceutical ingredient. This is the one which is the drug, which is the active molecule, which is acting against our target. So this terzepatide is the one which is called as drug substance. So this is the drug substance, and you need your drug substance to be dissolved in a particular solution, or if in case it is a oral thing, like then you need something to mix it with, so that you have a form of tablet, so that it is stable, so that it gets absorbed in your body, so it gets, mixed with your bloodstream or whatever when you have an intravenous injection or something like that. So those extra things that you add, that becomes excipients or a formulation, and the combination of formulation and active pharmaceutical ingredient is known as drug product. So the final marketed thing is drug product, but the one you start with is a drug substance, you can call it. Okay, and drug product can have combination of drug substances as well, or different dosage of drug substance as well. Like over here you see this is Monjudo, which is 2.5 mg per 0.5 ml of strength of formulation. But there are other formulation which is around 12.5 mg or 7.5 mg per 0.5 ml formulation are also available. So this is the difference which I wanted to mention. Uh, Also, as I was telling you, like peptide is a very new field, so there are still research ongoing in like what kind of guidance should be there, or guidelines should be there, which companies or researchers should follow to have a drug kind of peptide. So various agencies, European agencies or FDA, they come up with draft. This is a draft guidance. This is not the actual guidance. These are the draft guidance, and we hope to see the actual guidance soon in near future, as the number of peptide drugs approvals are increasing. So soon we will get more clarity on these things. But these are available free of cost on the various agency websites. So I would request like people to go through it and understand how the peptide drug development works. What are the requirements? Like it's not like you just develop a peptide which is a good binder and you can go ahead in the market or send an application to FDA for the approval. But there is a huge understanding required before actually submitting an application. Also, there are various quality guidelines which we need to follow, and these quality guidelines are very strict. But once you follow, they make the life cycle of the drug very easy, or the life cycle of a company as well very easy. So There are various quality guidelines, which we call ICH quality guidelines. There are you can see Q1 to Q14. So there are sub guidelines as well, and every company follow these guidelines if they want to have an approval. So yeah, like this is what I wanted to tell you, like how you can have a drug which is a peptide, how you can have it from market to from lab to market, and different guideline, different procedure, different analysis, different understanding that you need. And it's not just a chemist, it's not just a biologist, it's not just a bioinformatician. It's a cumulative effort of each and everyone in the field of whatever science you call it. They all come together for a success of a drug in the market. So it's really a huge effort which everyone puts in. And finally, we get to see something like terzepatide, which was approved by Lilly, which is a drug from Lilly. So that is all. Finally, I just want to tell you that so far, whatever knowledge I shared, actually I gained while I was working in different positions in different institutes, learning, understanding. Still, the journey is ongoing, and I'm happy to connect with people over LinkedIn or via mail or something like that. So, if you have any question, if you have any suggestion, please let me know.
But Dr. Hitesh, I have a few questions for you. You have a broad skills from your academia to industry. So have you struggled with this academia to industry transitions? So Dr. Hitesh, thank you.
So this is a thing like many people talk about in academia to industry transition. Indeed, it is difficult. I won't say it is easy or it is straightforward or something like that, because still, uh, like if we specifically talk about PhDs, they focus on a very small niche or a small research topic, but they have to understand that that small niche of is actually a field where they can learn a lot. Just try to focus on our science, but we can actually learn various things like project management if we talk about it. So actually, PhD students are managing a project, like starting from identifying a question, a research topic, and then ultimately solving it with various experiments, various analysis, documenting everything. So it's a whole management which everyone follows, but we don't consciously focus on these topics. Like
Yes.
we mostly focus on whatever science is going on and research or understanding. So I've seen many people struggling with communication as well. Like they are very good scientists, they are very hardworking, but they don't communicate well, they don't present well, they don't talk well. So these are the challenges that individual has to focus on. And once you tackle all these things, then I suppose it becomes easy, because ultimately in industry as well, you're taking up a project, you're trying to solve a problem. The working style can differ, but the way you work or the objective which you work on remains the same. So though it was difficult for me, because like I started searching for my job during COVID time. So like when I was in my postdoc in EPFL, then COVID started, and unfortunately I could not continue my what do you call contract. So I had to move back to India, and then I had to look for jobs. So during that time also, like I was trying to find some academic position, but now I see it as like I was fortunate enough that I did not get any academic position, and I did my transition into industry. Because what I feel like, in when you stay in academics, you just learn certain things, or there is a limitation what you learn. But when you change your like as you change from academia to industry, or if you change something, then you are exposed to different objective, different understanding, different types of people, and then you start you know uh, changing your own style, changing your own understanding for those things.
Yes, Dr.
So
Hitesh.
that transition is difficult, but
Yeah.
but there's such as nothing is easy.
Okay, thank you, Dr. Hitesh. So we have a questions. with the Dipesh Joshi. So Dipesh Joshi want to ask that what is the future and growth for antibody drug conjugates and the antimicrobial peptides based project in pharma industry? And so
peptide. Both of these are really hot topics. Antibody drug conjugate, why is it so? Because see, antibodies can specifically bind to a particular target or a particular protein, and you can have a targeted effect. Like in many cancerous condition, uh, antibodies are the choice of drugs. But being conjugation with small molecule or something like that, they gives an extra advantage, added advantage. And many industries are actually working for antibody drug conjugates, like developing their molecules. And we have seen, like, though I have not presented in my slide the number of ADCs getting approval every year, but we see every year one or two ADCs getting approval. So indeed, people are working on antibody drug conjugate. Talking about antimicrobial peptide is a hot topic. Many antibiotics are now, you can say, are facing the challenge of XRD or MRD. And these antimicrobial peptide pose or present as an alternative candidate for these challenges. And many a time people say that these antimicrobial peptides can be a super drug or something like that against various antibiotics. So various against various pathogens or bacteria or something like that. So many companies are actually, or many academic institutes also are working again for developing antimicrobial peptide. They are developing cell-penetrating peptide. They are developing various, biophysical studies they are performing. So to have a antimicrobial peptide or something like. So yeah, like these drugs are evolving, and these category of drugs are evolving. So.
yes. Oh yeah, as I have a few questions in starting of the your slide that what is the rules and regulations for under for choosing a drug. But you already mentioned it in the two slides. There's some FDA rules for choosing a drug. So and the thing is that drug substances and drug, what is the drug difference between a drug substances and drugs? Are two different things, and you define it very well. That is, a drug substances is a very different from the drug that we are we are taking or prescribed in the medicines. So very informative it was for me, and very new things were for me as well as for my participants also. So the second question is I want to ask, it's although very on a background of your personal, that is, what is your personal growth in terms of when you worked in India or you worked in a German company? So what is in terms of your personal growth? How do you find yourself as in a terms of your personal perspective?
Okay, so like I have worked in academia in India and academics in Europe, and then again in industry in India and industry in Europe. So I have both the experiences. One major thing, like which I felt, was the availability of the resources. There are like, because these are developed countries, they have money, they can invest. So the academic institute that you found over here will be having better resources. I'm not saying India is anywhere lacking or anywhere behind. We are doing very great in terms of competition. But what you, like, first glance, what the differences that you observe is this, that the resources are very much available in Western countries. Also, the working style. Generally, what I have observed, that the kind of work environment that you observe in India is quite different than what you observe in. I'm talking about European countries because I don't have experience in US or America or Canada, but I'm specifically talking about European countries. So that work culture is very much different. Pay scale, again, it's a huge difference between the
Yes,
financial
definitely.
growth that you observe in India and that you observe in European countries. So yeah, these are the few things which you find distinctive. But more or less, like, everyone is working for science. Everyone, like many Indian companies are there, and I would say the market is different. Like, uh, I won't say many discoveries are happening in India, but they are developing processes. They are working on, like the companies which I was working on was on generic peptides. So India is a huge market where we have a market for generic drugs. Also, if we talk about CRO, CDMO, these are like kind of companies who provide services. They don't do their own research, but they help other companies grow their business. So India is a huge market where you find CROs, CDMOs, CSOs. So these are the things. But in Western countries, you mostly see people who they perform mostly research and all. They do funding on these things, and they outsource to CROs. So India and China are the markets where you can outsource your activities. So again, like when someone works in CRO, CDMO, and when someone works in discovery, these two are different altogether. Like the objective becomes different. The understanding or the discussions are different. They are not the same. So it depends on what kind of path you are choosing. It is very subjective, and also we cannot directly compare uh, Indian companies or European companies, because again the size of the companies are different. The investment coming in is different. The market they are targeting, mostly Indian companies target US market,
Yes.
and European companies target US as well as European market, and then they have separate thing for the Asian market. So again, the objective is different. And why I'm talking about market? Because the regulatory requirement for different markets are different. So if you start talking about regulatory requirements, then India has different regulatory requirements. Like our CDSO, they have different regulatory requirement in India.
Yes.
China has its own, Japan has its own, Europe has EMA, US has FDA. So
I must say thank you to you for giving your fabulous speech that you already presented in your slides, and as well as want to thanks to my all participants who continue with us to hearing your question, to hearing your answers and your presentations. And thank you to, again, your Dr. Hitesh. I think on this topic we will discuss later with a different title with you. So thank you for all. I think the attendees don't have any other questions. So if you want to give any feedback to me or to our Neomaticians, so please welcome. Please give any feedback you want to convey to my participants or to Neomatician as well.
Yeah, so thank you, Neomatician. Like it's a good initiative to have a webinar series, and I look forward to other speakers as well. And thank
Dr. Hitesh, at last moment, we have one question for Dipesh Joshi that after PhD, what is the expectation salary in Indian company if we want to shift to industry? So this is a kind of person who already gained their PhD and wants to move to the industry. So what is?
So pay scale, I would say, like different companies offer different. They have their own pay scale. I won't tell you a specific number
Thank
because
God he
it
didn't
can
ask
vary.
about your pay scale.
Yeah, but it's a decent pay scale that you get in Indian companies, and you can grow with your skill sets and everything. Uh, uh, and there are room for negotiation as well. So like if someone has a specific point, then I can discuss personally. Like if someone is undergoing any recruitment procedure and want to understand, like there are like companies offering this much, then I can discuss in person.
Sure. Okay. Thank you, Dr. Hitesh. We will meet it again.
Yeah. Okay. Thank you very much, and thank you, Neomatician, and thanks, Preeti, for the organizing. Yes, thank you.
Thank you, Hitesh. Thank you. Thank you, all the participants. I hope please keep in touch with us. With our websites and on our social media pages, so that you can keep updating yourself regarding our ongoing webinars on the courses. So thank you all.